Thank you, Professor. Good afternoon. Thank you, Professor Roos, for kind introduction. And I thank the organizing committee to invite me here for this talk. And a lot of people have done quite a bit of work in non-thermal area, and there are many new technologies for non-thermal food processing, food preservation. So we started doing some work with atmospheric cold plasma about 7-8 years ago. So in a way, I'm probably latecomer for this cold plasma, but I'll give you some overview, some insights on this. Okay, so give a quick introduction, some information on methods to generate cold plasma, microbial inactivation. We have done some work with E. coli inactivation in apple juice, apple cider. Also, we have done some work with quality of the cold plasma process juices, ketchup juice and apple juice, and then conclusion. Probably you all know the cold plasma is the fourth state of matter. It's basically ionized gas which contains ions, electrons, and excited atoms and molecules. It's produced when energized. Electrons create reactive species from neutral gas atoms. Also, it has application in food preservation, as I mentioned, and also we can modify food components without heating. Some of my previous work has been on modification of proteins and other aspects of food, but this talk is mainly on food safety. So if we look at the characteristics of cold plasma, I can see on the top, and let's see if this thing works. The first thing you'll notice is reduction in pH. pH is used depending on the product you are treating with. It produces ozone. Ozone is quite obvious. You also get light, UV, infrared, again depending on type of devices. You get charged particles, electric field, and reactive oxygen and nitrogen species. So those are the two main things we look at. And these are excited species. I just show a quick picture of few type of devices. There are a lot more types. You can see on your left side, it's dielectric barrier discharge type plasma device. Then there's a plasma jet, corona discharge, which is mostly for little higher temperature applications. We have microwave type discharge. Nowadays, a lot of people use dielectric barrier discharge type unit, but others are also available. This is the type of device we have in our lab. Basically, we put all these components together. We bought these from Plasma Jet, and you can see on the top, that's where we generate the plasma, but at the bottom shelf we have the generator to create the high voltage and so forth, the cooling unit, the chiller, and in between that little cylinder hanging in there, that's our treatment chamber. So that's where we can put the samples and then bring the plasma. That's the back side of the unit, so you can see we can control all the airflow of water and measure whether oxygen, nitrogen, air, whatever gases we are passing through. Here is another example of cold plasma people have used for in-package applications also to increase shelf life. So you can see there are different type of applications, like in food waste, create nutrient chains, I mentioned enhanced self-life, delayed poor days, and so forth. So there are many other applications of this cold plasma. Let's look at just a quick mechanism. The cold plasma can have either biological, physical, or both. So there are both ways of inactivating microorganisms. In biological inactivation, the cells can be damaged by UV radiation, which is produced by cold plasma, and that changes the DNA of the cells. Also, there is lipid peroxidation, which is UV light, in addition to reactive species, charged particles, and other oxidative species will change the lipid peroxidation. That changes the cell structure and the bacteria dies. So there is change in protein. The UV can promote cell surface erosion, which is a synergistic effect, and also the physical inactivation, where the cells are damaged by electrostatic disruption and electroporation. Here is on the right side some pictures. You can see undamaged cells and the damaged cells. That's the morphological change after treatment with cold plasma. And basically you can see the active species will cause the oxidative damage of the cell. Also, it can damage the protein structure of the cell or electrostatic disruption. There is another example where you can see the efficacy of plasma-activated water. So there is quite a bit of work going on, not directly with the plasma application, but we create plasma-activated water, where you take the water has unique property, and you use that water to sanitize things, or use for cleaning bacteria. So here is an example of, there is left side, the control, then plasma-activated water treatment for one kind of microorganism, then stuff, ARIA cells, and you can see in each case, you see the cells shrink or damage after the treatment with plasma-activated water. What are the factors they affect? Basically, we did some experiments with E. coli, so in that case, the prolonged exposure enhances the microbial inactivation, which is well known with any treatment. If you expose for a longer time, you get more inactivation. Also, gas composition. Different gas mixtures affect the bacteria differently. People have used helium, argon, oxygen, and nitrogen. Only thing is, argon is very expensive, helium explosive. So most people try to use combination of oxygen, nitrogen, or air is the cheapest. So a lot of new applications you will see is mostly with air, and that's what we use to simulate air by mixing oxygen and nitrogen. Food surface characteristics, if you are treating like produce, or some people have used different steel surfaces, surfaces of like conveyor belts and so forth. So those surfaces have certain characteristics because of their porosity, moisture content, organic matter, and so forth. And then device configuration, because depending on the electrode designs, many electrodes, I showed you, they are flat electrodes, they are needle type electrodes. Now people are making curved electrodes. So there are different kinds of electrode designs also. As an example of treatment of coconut water, there they look at total viable counts, and they decrease as the treatment time decrease, which is, treatment time increase, and they also restored. The only thing you'll notice, some of these treatments as you increase the storage period, the microbial counts will start increasing. And that's a classical example of injured cells. So if there are cells which are injured in the treatment, they could regenerate later on. And that's why one has to do experiments whether you keep the product refrigerated or keep it at higher temperature, depending on what the kill rate is and how you store it. So in this example, they store, they start seeing increasing the cell count. Here is an example of decontamination of lettuce using plasma activated water. And again, you can see this one, y-axis has actual the viable that counts. So you can see at different times, 5 minutes, a less viable count, and there's what they did, they treated the liquid water, put the pieces of leaves, and agitated and non-agitated. So that's why you see two graphs, agitated versus non-agitated. Of course, it's obvious if you agitate, you get more intimate contact, so you get higher reduction, less survivals versus do the non-agitated. And also did gram positive, gram negative. So on the right side, you see the top one is the gram negative bacteria and bottom one is gram positive. So you can see there is a more effect on the gram negative than on gram positive. So again, you have to experiment see because the plasma does more inactivation of gram negative than it does for gram positive. The same way here they use samples to spread E. coli on petri dish and then use the gases to treat. And you can see as the treatment time, in this case, increased, their temperature increased though. So we're not sure whether the effect, synergistic effect of temperature, though the temperature increase was not very high, but still there is some increase in their temperature. They did not control the temperature very well. The units we have, we control temperature within 2 degrees Celsius. So temperature doesn't change from room temperature to maximum 2 degrees. Also, a study which simulates like equipment where they use the steel coupons and the conveyor belt where they use the plastic or coupons, and you can see and they put those coupons at certain distance from the plasma application for certain time. So that's what on the x-axis you see 3 centimeter, 60 second to 7 centimeter, 60 second. And in this case, as you have increased the distance, you have less inactivation, which is also kind of obvious that, but they had more inactivation on that PVC coupons because of the roughness of the surface and so forth than on the steel. So they mentioned the surface also has effect if you are treating the equipment or conveyor belt. It's the same study. You can look at it. It's the same treatments, but in some cases, it's not a very clear conclusion. But you can still see that stainless steel inactivation was not as high as they had on the PVC inactivation. The work we started, we also used E. coli K12 as a surrogate because in many food plants you don't want to bring pathogenic E. coli, so we were trying surrogate in the lab as well as the pathogenic. And we wanted to compare those two for apple juice. Also, we wanted to see if acid adapted versus non-acid adapted K12 would behave differently. And then we also tested the pathogenic E. coli 0157S7. And it's just a few slides I picked up. There's a whole PhD work, and we don't want to bring too many things, just a few key slides. So in this case, see, we used what we call simulated air, which was combination of 80-20, 80% nitrogen, 20% oxygen. And also we call modified air, where we had 90% nitrogen, 10% oxygen. We did not want to add more oxygen. We had done some experiments early on because it creates too much ozone. Ozone, people have done experiments with ozone quite a bit, and it creates bleaching effects, and there are so many other quality effects, flavor effects, and so forth. So we stayed with similar to air, and you can cut back on oxygen, add more nitrogen. So, and we also wanted to test if the growth medium where you grow and test things will have any effect. So we used Macanthi agar and also took soil agar. So you can see in most cases, other than the combined, which is 90% nitrogen, 10% oxygen, has little delayed effect, not as effective as the air 80%. In fact, in 2 minutes, I got quite a bit reduction with simulated air. The same thing with the modified gas, it took 3 minutes. So in 2 minutes versus 3 minutes, they all got to the similar reduction. And you can see on the bottom row, so this is the non-acid adapted. Acid adapted, so those took about the same, maybe 150 seconds. And the difference between the two kinds of gases is not that clear, because of so much high standard deviation. But it's still like basically 2.5 minutes. We can get very good result. This is the E. coli 0157S7, and in this case you can see the simulated air. We get reduction in 90 seconds, very quickly. So in a way I can say my K12 surrogate is more resistant than the pathogenic E. coli. So if I can process properly with the surrogate, it should be safe for the pathogenic E. coli. Here is some example of storage. And after the treatment, we stored the samples at 25 C. And also, I think we had stored some at 4 degrees C, you can see. And we have both kind of medium to grow them again. And you can see that at 25 C after 24 hours, we still have quite a bit reduction in both cases. And that tells that even at room temperature, these cells were dead. They did not reserve there. So we did not have too many of those injured cells in that case. And even the post-process storage, same way you can see here. We started with almost 8 log. We got down to 3, and in the simulated air to 2. So simulated air seems to be more effective. And I think the main thing was I was getting a little bit more ozone in the simulated air. So it could be the effect of that ozone. Okay, Same stuff I think is showing with the arrow. Then I also tried on spores. And now these results are totally different. You can see there is hardly any change on the spores, yeast molds. That was kind of a little surprise that this cold plasma was very effective on bacteria, but not as much as on spores. With time it looks like coming to close. So you can see here basically the actual ozone concentration in all the juices was very low, below 0.2 ppm. In apple cider, deionized water, we saw a big, pH change, pH drop. And you can see a lot of other things in there, which students measured. We didn't see too much other change other than the change in the pH, and of course some changes with the ozone. Okay, this stuff, same way our results confirm what other people have done. You can see the bacterial cells after treatments, they look shriveled, shrunken, they leak, so they are inactivated. And we also did a little bit on spectra, what's coming out of that device. And you can see this spectra is basically UV range. So we are getting quite a bit UV coming out of that device, which also has a antimicrobial effect, and that also caused the inactivation of microorganisms. So we can conclude that our K12 is a pretty good surrogate for E. coli 5787. Also, the growth media did not make much difference. The HCP protected most of the bioactive compounds, which I did not show here. We had done quite a bit measurements on those. We also did measurements on volatiles. That's a whole different publication by the student. And some volatiles were decreased, some volatiles look like increased a little bit, depending on the type of the volatile. And that's what these two are. So there's some increase, some decrease. We did not do any consumer acceptance. The main reason was the whole device was in bi-level 2 lab. We cannot ask people to try anything we make there. So that's why we stopped there. So maybe somebody someday can use this technique and do some consumer testing. So I'll just stop here. Thank you.